Well, thank you, Xiaowei, for that fantastic talk, and you can see that it fits really perfect into the idea of convergence. So our next speaker also is very convergent in science and physics. Dr. Daniel Larson is from NCI. He's the NIH Statman Investigator in the Center for Cancer Research. He's also head of systems biology and gene expression in the National Cancer Institute, and he's a pioneer in vivo single molecule studies. And he's also a recipient of the Presidential Early Career Award in Science and Engineering. So today he's going to speak to you about understanding transcription and splicing heterogeneity in cancer progression.
Great, thank you. Is there a laser? Well, thank you. Well, thanks very much to Phil and Susan for the invitation to speak. It's an absolute pleasure to be part of this symposium. As Angela mentioned, I'll be talking about cancer heterogeneity, and I thought I would start with a particularly striking example of that. This is a field of MCF7 cells. This is a mammary carcinoma cell line. And you're looking at a single molecule FISH image, which is very similar to what Xiaowei showed. And this gene, this message is TFF1. It was one of the first, I think the first estrogen responsive gene ever cloned by Pierre Chambon. And as you can see, there's a tremendous amount of variability. In fact, it's one of the most highly variable genes we've ever seen in human cells. The most likely number of RNA per cell is 0, and some cells have over 200. You can also, this same phenomenology can be viewed by single-cell sequencing. So this is essentially the same observation writ large. This is a recent paper in primary breast cancer cells. These aren't TADs. These are just clusters of gene expression profiles in these cells. And these are all the carcinoma cells. So again, you can see this variability, this cohort of cells which have different gene expression patterns even when extracted from the same tumor. Now, when I see data like this, some of the questions that come to mind are how much of this variability is genetic? I think over the last 30 or so years of cancer genetics, we know that a lot of it is genetic. How much of it is non-genetic, and Xiaowei alluded to some of that. But also, how much is static and how much is dynamic? In other words, if we looked at these cells a short time later, would they show a radically different gene expression profile? And then finally, the question that the whole field is asking is how much of this heterogeneity is functional, in other words, leading to true biological subpopulations with different phenotypes, different roles in cancer progression, and how much of it are just fluctuations that may be biologically unimportant. So during this talk, I'm going to try and at least address this last question. So I'll talk about the underlying causes of a lot of this heterogeneity, and then turn to splicing factor mutations in cancer and present a model for how we think these mutations are working. And one of the techniques you'll see throughout the talk is this ability to visualize RNA in living cells. This is a fairly old technique now. It was originally developed by Rob Singer in the late 90s. And it consists of inserting into the gene of interest a DNA cassette which codes for RNA hairpins. When these hairpins are transcribed, as you see here, this phage coprotein, which is labeled with a fluorescent protein, binds the hairpins, and now you have a fluorescent RNA. So this is an example of what this looks like when you look at human genes as they're transcribing. This is, again, the nucleus of an MCF7 cell. And what you're seeing in these movies is the real-time transcription of this TFF1 gene. So this was done by CRISPR-Cas9. We've labeled 3 of the 4 alleles in MCF7 cells. And each one of these flickering spots, which this is a movie which lasts over 14 hours, is a reflection of this underlying process. So what you're seeing is a polymerase will start transcribing. It begins to make the RNA, shown here. Once that stem loop cassette region is transcribed, now the hairpins are available for binding by the fluorescent coprotein. Now you have a fluorescent RNA at the site of transcription. Eventually, this polymerase proceeds down the body of the gene. It releases, and then this polyadenylated mRNA is released as well. Now, the thing that jumps out to me when I look at these images is that this gene, TFF1, is perhaps one of the most upregulated genes in human females in response to estrogen stimulation. Yet, when you look at it in this way, you can see that transcription is actually a really rare event. If we look at this in greater detail, and image at very high speeds, we can see, again, the nucleus of the MCF7 cell. We see these bright spots, which are sites of nascent RNA. This is where RNA is being synthesized. And we also see these RNAs diffusing around in the nucleoplasm. Here's an example of a cell which is uninduced with estrogen. So these are the quiescent state of this cell. And as this movie loops at the 14-hour time point, you'll see when estrogen is added, right about here. All 3 alleles turn on, and then they quickly start to show this independent behavior within the population. So we can track these cells over long periods of time. So this is essentially 14 hours of single molecule imaging. And these are just 3 different cells with the activity of each allele false colored in either red or green. So here's an example of a cell on the upper panel, which shows a lot of activity. Both alleles are active for almost the entire duration of the experiment. The cell in the middle panel shows an example of one allele which turns on and then turns off for almost 12 hours and then turns back on. Meanwhile, the same nucleus has another allele which shows activity. These are these green bursts. And we also see incidences of complex spatiotemporal correlations between alleles. So I'm not going to go into any more detail about this, but I can leave you with the conclusion that these alleles are not independent. We can collate all of that data into a histogram and a heat map. And so on the heat map, on the y-axis, these are all individual alleles. This is the raw data. There's no image processing or bleaching correction or anything. And this is the frequency histograms of this data. So I think you can see that the most likely time for this allele to be off is around 15 to 20 minutes. But alleles can also stochastically switch off for 12 to 14 hours, while other alleles in the same nucleus are still active. And I'm just showing you a few dramatic examples, which you see near the bottom of the heat map. So what that means is that since this off time is actually longer than the half-life of the RNA, that the cell is going through periods of stochastic monoallelic expression. Now, I want you to remember that because that's going to become important later when I start talking about splicing factor mutations. We've also developed methodologies to look at transcription and splicing at single alleles. And this is just a 2-color variation on this same system. So in this case, we have the intron labeled in red. We have the exon labeled in green. When splicing occurs, we remove this red signal, and we're left only with the green. And here I'm just showing you a movie which summarizes this process. It's fantastic to be here, as Tyler said, on the 40th anniversary of the discovering of splicing, but he neglected to mention my favorite part of that paper, which is that splicing was essentially discovered by microscopy. So I've always had a special affinity for this process. Okay, so everything we've done in the past has been done with targeted insertions or model genes. We've now developed the capability to look at thousands of genes using this single molecule live cell approach. And we're doing that with an old technique with some modern innovations. So this is the classic gene trap approach. And you've probably heard of enhancer traps or intron traps or exon traps. This is an intron trap. And basically, you make a cassette which has a splicing acceptor in this lentivirus. There are markers for finding these cells, like RFP and blastocytin resistance. This piece of DNA goes randomly into the genome, but if it goes into an intron, It's spliced into that upstream gene, so it doesn't have its own promoter. It only gets expressed when it goes into an intron, and now you splice into this splicing acceptor. We cut out all the pieces that we don't need, including the resistance marker and the fluorescent protein, and in the end, we are left with a gene with these stem loops in introns throughout the genome, which we can then map by deep sequencing. And as expected, most of these insertion sites are in introns, although not exclusively. So in this case, we've isolated and subcloned about 1,000 introns with MS2 stem loops and about 8,000 introns with PP7 stem loops. So I'm not going to show you all 8,000 of those genes. I will show you a few as we sort of hone our high-throughput imaging pipeline. Here's an example of SLC2A1. The gene is going from the right of the screen to the left. This browser track is actually nascent RNA, so we're just using the Uli-Schiebler chromatin-bound RNA fraction. And this is where the stem loop cassette inserts. Now, most of our stem loop insertions seem to be in long introns, which is perhaps not surprising. If we now look at the dynamics of splicing, this is what we see. So this is, again, a frequency histogram on the y-axis. Oops, and this is time in minutes. Sorry for cutting that off, on the x-axis. Now, some introns splice very quickly, in less than a minute, but most introns splice on around 10 to 15 minutes, although some introns take over 60 minutes to splice. Now, to put that in perspective, a polymerase moving at what we think is the average polymerase velocity would reach the 3' splice site, the splice acceptor, at that time. It would reach the end of termination at that time. And so everything that happens after this transcription termination site is on introns, which are retained for long periods of time at the site of transcription. So the on times report on transcription and splicing dynamics. The off times are the signature of regulation. So when this gene turns on and off dictates how much RNA is in the cytosol. So again, as we saw with that estrogen responsive gene, we have many off periods which are on the order of 15 to 20 minutes, but we have some off periods which are on the order of 6 or 7 hours. And I think it's fair to say that we don't really have any idea what's generating these long time scales. As we start to accumulate data on many genes and many introns, some patterns start to emerge. So I'm just showing you 3 different cells, 3 different introns. You can see some genes like row A, which fire quite a lot. You can see some genes like ERRFL1, which fire hardly at all. But these intron processing times are remarkably consistent from gene to gene. So here I'm just showing you 7 or 8. Now we've done 20 or 30. And these cumulative histograms are almost on top of each other. So the time to splice these long introns seems to be the same, whether the cassette is located close to the 5' splice site, close to the 3' splice site, whether it's a first intron, whether it's a last intron. And we are trying to understand where this comes from. And again, this off-time distribution varies from gene to gene, because this is the signature of regulation. Some general principles are emerging from these studies. So I've just showed you our work in human cells, where we measure this burst size of around 3 RNAs per burst. Our work in yeast returns a similar burst size of around 2 RNAs per burst. And then work in flies, this is for the Eve stripe 2 enhancer, around 5 RNA per burst. So in this case, I think it's clear that most of the noise or the variability in human gene expression doesn't come from many RNAs being initiated at once, but it's these long and variable periods between when synthesis occurs. So how is this relevant? I'd like to turn now to a different topic, which is trying to understand how these dynamics are perturbed by splicing factor mutations which are found in cancer. Now, around the time that I started my lab, there was an outpouring of data from the TCGA and other type studies which revealed mutations in the general spliceosomal machinery. Now, people who have worked on splicing for many years would say, well, we knew about splicing factor mutations in cancer, the work from Adrian Krainer and others. I would say the surprise from this data, and it truly was a surprise, was these were mutations in spliceosomal components which were thought to splice almost all introns, 90% of human introns. So why this had disease-specific or pathway-specific effects was very interesting. So if you take my personal 4 favorite splicing factors, U2AF1 being the first, SF3B1, SRSF2, and a minor spliceosome component, ZRSR2, add up the mutation rate, you can see along the x-axis all these different tumor types and the abundance of mutations. So for some cancers, this is uveal melanoma here, and diseases such as MDS, splicing disease is the major molecular phenotype of this disease. Now, the one that caught our attention was a hotspot mutation in U2AF1. So U2AF1 is involved in 3' splice site recognition. There's a cartoon of the protein structure. You have a zinc finger domain here. You have a second zinc finger domain here, this UHM domain. And all of these mutations were in this first zinc finger, more or less. And this is a mutation of a serine to a phenylalanine. Now, and it was always heterozygous. And you don't have to be a cancer geneticist to recognize that this is a signature of an oncogene. So it's a hotspot, heterozygous, probably some sort of gain of function. And to bear that out, work from Tim Graubert, who is here in Boston now, showed that if patients who had this mutation had a much worse prognosis of progressing from MDS, myelodysplasia, to AML. And that's what this data is here. Angela Brooks and Matt Meyerson demonstrated that these splice site mutations showed a slight alteration for the 3' splice site sequence, although I wouldn't say it was black and white. There seems to be a preference for a C at this just upstream of the AG dinucleotide versus a T. But again, some cells have this. Some RNA show this behavior, some don't. Some patients show this behavior, and some don't. So it's not a hard rule. We had shown that the molecular phenotype of these splicing factor mutations is that splicing seems to occur much more slowly. In fact, for some introns, it's slow enough that you no longer have any co-transcriptional splicing whatsoever, and splicing is entirely post-transcriptional on RNA, pre-RNA diffusing in the nucleoplasm. Now, what is the physiological effect? To ask this question, we turn to lung. So this is human bronchial epithelial cells. These splicing factor mutations that occur around 6% of lung cancer patients. And starting with TALENs and then finishing with CRISPR, we made the following cell lines. We have the wild type cell line. These are a non-transformed, immortalized human bronchial epithelial line. We have the S34F mutation at heterozygous levels. We never achieved any homozygous clones, which again suggests that as a homozygous, it's lethal, and you don't see that in the patients either. And then we rescued this mutation by removing it. So for the rest of the talk, I'll talk about these 3 different comparisons: the wild type, the mutant, and the rescue. Now, I think I'm speaking for many people in the field when I say these are not strong driver oncogenes. If you put this mutation into mice, they don't get leukemia, they don't get lung cancer. In fact, if you didn't have the blood malignancy data, you might even be hard-pressed to call these as driver oncogenes in solid tumors. We see splicing changes. Most people see splicing changes. The one phenotype which has borne fruit for us is this observation that cells which have this mutation show damage resistance. So here's an example where we hit cells, these lung epithelial cells, with 20 gray radiations. This is a whopping amount of radiation. It's approaching clinical doses. And if you count these gamma H2AX foci and generate a histogram over around 10,000 cells, you see the most likely number of foci per cell at this time point is around 40 or 50. However, in the S34F cells, there are a population of cells which never show any detectable gamma H2AX foci. So as far as we can tell, they are not responding to damage at all. And this phenotype is completely rescued when we just mutate this one nucleotide back. Now, in data I'm not going to show you, we know that these cells are still getting damaged, so they are getting breaks, but they're not responding. But more importantly, these cells, which are now hit with 20 gray irradiation and have shattered genomes, live for over 2 months in culture, whereas wild-type cells die within a few days. Now, you are probably thinking, well, that there's no way these cells are proliferating, and that's true. They are in a senescent state, so they're not dividing, but they are metabolically active. And because they are in this senescent state and not proliferating, though, they are secreting other things which can have an effect on surrounding cells. And one of those is IL-8. So this is interleukin 8. So this is an ELISA study, and this is just a single experiment where you see what should be the response of IL-8 in response to damage. So at time 0, this is before radiation, we hit with 20 gray, IL-8 shoots up, and then it starts to go down as cells start to die away. A haploinsufficient shows the same behavior. The S34F shows this extended period of IL-8, and then again, we completely rescue that. More importantly, even before damage, we see almost 20-fold upregulation of interleukin 8 secretion. So this is the same data now with over 6 biological replicates, 2 technical replicates each, so it's 12 replicates. And again, you see this sustained, secretion of IL-8 in some cases, many months after a lethal dose of irradiation. Just for completeness, the only other cytokine we see changing is IL-1 alpha. Now, if we take human cells, in this case, H441 cells, and we take this mutation and we correct it. So this is a human cell line which normally has the S34F mutation, isolated from a patient. We correct this S34F mutation, and we see these IL-8 levels go down. It's not the same magnitude of change that we see in our isogenic lines, which is shown here on the left. So this is IL-8 in the isogenic lines. This is all before damage, so we see wild type, we see a haploinsufficient, we see this S34F, and then again, we completely rescue that. But it is in the same direction, which is encouraging. So coming back again to this idea of rare cells and what they do and whether it's important, we have now started imaging. So this is a gene-edited line which expresses an IL-8 GFP. This IL-8 is actually secreted with the GFP, which was somewhat remarkable. And on the left is the wild-type U2AF1, so this is the non-mutated case, and wild-type interleukin 8. And so what you'll see are cells that look, I think there's even a dividing one right here. This is the basal level of IL-8 secretion. This is how much IL-8 these cells are normally putting out. If you introduce this S34F mutation, now we start to see cells that look like this. As the movie loops again, you'll see the cell sort of elbowing its way through the epithelium. I believe these bright punctate spots are vesicles of IL-8, some sort of secretory trafficking vesicles. But again, it's maybe more overall in the cells, but certainly these outlier cells, which just have a lot of IL-8 production. If we now do a gain of function mutation, so this is a mutation in one of the 3' splice sites of IL-8, we can now make these cells just pump out all kinds of IL-8. So to put that in perspective, this is a trans-acting mutation which changes IL-8. This is a cis-acting mutation in IL-8, which dramatically enhances this phenotype. So the question we had is are these small number of cells that are showing this aberrant behavior, is this enough to give you cancer? Is this enough to transform cells? So what we did is we took media from these cells and we put them on primary mammary epithelial cells. This is an immunohistochemistry of E-cadherin and fibronectin. So these are markers which are commonly looked at to understand EMT. This is the intensity of fibronectin in one of those cells versus the intensity of E-cadherin in one of those cells. And this is the normal behavior. This is what you expect for epithelial-like cells. If you just dump IL-8s on these, now you see this signature of the mesenchymal transition. You have high fibronectin and low cadherin population. If you take media from the wild-type cells, you can't achieve this sort of change. But if you do take media from these S34F cells, these HBEC cells, now we see a population of cells which shows this EMT phenotype. So I'd like to conclude with just leaving you my closing thoughts on heterogeneity in this context and how it is working in splicing factor mutations. I hope I've shown you that this process of noisy, as it were, transcription and splicing leads to a very dynamic, heterogeneous population of cells. So you have cells which have different amount of RNA and different isoforms of RNA. In cells which have a lot of this S34F mutation, which again just happens stochastically, you see heterogeneous splicing changes, R loops. I didn't show you any of that data. Interleukin 8 secretion. These cells, when hit with high doses of X-rays, have enhanced viability, long-term senescence, and they show the sustained IL-8 secretion. And we think that this mutation, at least in this in vitro system, is really acting in a cell non-autonomous manner. So it's not the cells that have this mutation, but it's what they do to their surroundings. So these cells, for example, after senescing, will secrete these factors, these cytokines, and this is generally has a bad prognosis for the tumor. So with that, I will stop. I want to thank the people who did this work. This is work from Murali Palangat, Joe Rodriguez, who's a postdoc in the lab and an MIT alum, Yihan Wan, and all of the, some of the splicing factor work is done in collaboration with Harold Varmus and his postdoc Dennis Fay. That's mostly the mouse stuff, which again I didn't show. And we get occasional bioinformatics support from Rob Bradley at the Hutch. So with that, I will stop, and thanks for your attention.